Hello everyone, this is Andrea Cabibbe from San Raffaele Hospital. In this presentation, we will see what are the key uses of NGS for diagnosis and surveillance of drug-resistant tuberculosis. While novel and repurposed drugs like bedaquiline, linezolid, clofazimine, pretomamide, delamanid, and pyrazinamide are increasingly used in TB treatment regimens, rapid molecular tests to detect resistance to these drugs are lacking. This gap hampers effective treatment, especially in drug-resistant TB cases and for group A, B, C drugs, where TNGS or WGS represents currently the only comprehensive rapid option for susceptibility testing. Indeed, as we can see exemplified in these two rapid testing algorithms, currently there is no WHO-approved molecular diagnostics that exist to rapidly detect resistance to many new anti-TB compounds. And this results in providing treatment regimens despite being blinded to the susceptibility profiles of such drugs. Next-generation sequencing of M. tuberculosis provide comprehensive drug resistance profiling, informing the use of modern drug-resistant TB regimens, and also offering a suitable platform for future treatment changes. NGS overcomes the long turnaround time of phenotypic testing and the limited resolutions of the available rapid diagnostics. I now bring a couple of examples of the utility of the use of NGS for drug-resistant TB diagnosis. This retrospective study from South Africa evaluated the impact of WGS on treating rifampicin-resistant tuberculosis. Findings revealed that WGS could have led to more effective treatment regimens for nearly half of the patients by identifying drug resistance patterns not detected by the standard testing. Additionally, about a third of patients deemed ineligible for shorter treatment courses might have qualified with WGS insights. The study underscores WGS potentials to optimize therapy, reduce toxicity, and enhance outcomes in high-burden settings. This UK-based study assessed the role of routine whole genome sequencing in tuberculosis treatment decisions. Among 189 TB cases analyzed, WGS identified drug-resistant mutation in 12 patients, including 8 with isoniazid monoresistance. These findings led to timely adjustment in treatment regimens. Notably, WGS results were available in a median of 35 days, significantly faster than the 86 days needed for phenotypic drug susceptibility testing. This study highlights WGS potentials to enhance patient management by enabling earlier tailored therapy, particularly in detecting isoniazid resistance. In other examples, findings from two studies conducted in Italy revealed that WGS from early positive cultures would provide faster results compared to first and second line phenotypic DST, replaces the need of additional testing, and averted the need of phenotypic testing for more than 80% of cases, being susceptible to first line drugs. NGS is also a valuable tool to support public health actions. It is recommended by the WHO to support surveys and surveillance within the WHO Global Project on Anti-TB Drug-Resistant Surveillance. So far, 13 countries incorporated NGS into their survey protocols, and others are coming, particularly from high-burden settings. In the two examples on the right, whole genome sequencing in Djibouti's first national TB survey revealed that nearly 60% of MDR TB strains were part of transmission clusters, indicating recent and ongoing community transmission. Also, a national survey conducted in Eswatini between 2017 and 2018 showed that almost 60% of total rifampicin-resistant cases harbored the diagnostic escape RPB mutations affecting codon 491, and that transmission is ongoing. The country then adopted NGS into the national diagnostic algorithms to catch this outbreak strain. The European Centre for Disease Control coordinated the WGS-based pilot surveillance of RR-MDR-TB between 2017 and 19. That was then continued in the next years, showing that the use of WGS-based surveillance for monitoring MDR-TB outbreaks nationally and internationally is feasible and allows to elucidate cross-border transmission patterns in Europe, reporting the main lineages and clusters spreading in the region. This study conducted a nationwide genomic analysis of tuberculosis in Moldova during 2018 and 19. Among more than 2,000 culture-positive TB cases, 36% were MDR, with half of these in patients without prior treatment. Remarkably, 92% of MDR strains clustered into three major lineages, two Beijing and one Ural, each exhibiting distinct drug resistance mutations and geographic patterns. These findings highlight that ongoing transmission is the primary driver of MDR-TB in Moldova, underscoring the need for targeted public health interventions at sub-regional level. Come to the conclusions of these presentations. We are now well aware that ideally diagnostic development should parallel drug development. However, resistance mechanisms are not fully elucidated for TB drugs, even following regulatory approval, and real-life datasets are needed for deep investigation. Sequencing is an important diagnostic solution that can be used to interrogate a wide range of resistance-associated gene regions, but its use is currently limited to centralized laboratories. Existing NGS solutions would be currently blinded just to predominant drug resistance. WGS, on the other side, covers all drugs. The development of diagnostics, specifically for the detection of linezolid, bedaquiline, nitroimidazole, and pyrazinamide resistance for high-burden settings, should be a research and implementation priority.